Alrighty, good morning everyone. It looks like we've got about 62 attendees on here so far. Just a few housekeeping things before we get started. You should have the chat window open to you so you can pop any questions in. We'll really try to make this as interactive as possible. So if you have any questions as we're going along, we'd love to hear from you. And then we'll also have some time for Q&A at the end of the session. So please feel free to drop some questions in the chat here and say hello. Let us know where you're from and what you've got going on, what you're excited to learn. Something to kind of kick things off, just to engage all of you all joining virtually. If you have anything that you're really excited to learn about today, and that is just like pressing questions, something you came today and you're really hoping to learn, drop it in the chat with your name, who you're from, and hopefully we can address those questions as we go. I love to ask that at the beginning and get some engagement on this virtual platform that we spend a lot of time on these days. So if you're all here, it's possible that you're interested in emerging technologies and ag input manufacturers. So, you know, Biome Makers, 3-Bar Biologicals, and FarmQA, we're interested in biology and biologicals. So just wanted to welcome you all here and introduce myself and my fellow panelists. So I'll have them introduce themselves shortly, but my name is Meri Mullins. I am a regional manager for Biome Makers, so working with a lot of our clients, both farmers, retailers, and input manufacturers, Colorado north and south, and everything west. So that's kind of where I'm coming from, and I'd love to. I'm joined here today by Jane Fife and CTO at 3-Bar Biologicals, and Ben Munson at Senior Technology Specialist at FarmQA. So I'd love for Jane, if you could just introduce yourself really quickly, and then we'll have Ben give a quick introduction to him as well, and we'll kick things off.
Okay, sounds great. Thank you, Meri. Of course, thanks to Biome Makers for making this webinar possible today. Again, my name's Jane Fife. I'm with 3-Bar Biologics, and my background's I'm an engineer by training, and a lot of my research focuses on how to improve delivery of living microbe products, really reducing it to practice for farmers so that we can make these products work in the field.
Awesome, thanks, Jane. Ben?
He's muted.
Oh yeah.
Sorry about that. Yep, as I said, my name is Ben Munson. I'm a senior technology specialist at FarmQA. We started in 2015. I wear a lot of different hats with this role, so it's sales and it's support. It's also working with our partners like Biome Makers. And with that, I get to interact with a lot of growers, agronomists, and help them walk through our software and figure out how they can make better decisions based on the tool.
Amazing, thanks, Ben. So what we're going to talk about today is a handful of things. So in the biological space, there are a lot of questions that are emerging, right? There's a lot of attention on biostimulants, biologicals, just different ag inputs. There's a lot of questions around what can we do differently today in agriculture with the changing landscape. So what we're going to talk about today is really 3 primary things. We're going to talk today about research and development of biologicals and what Biome Makers is doing to support ag input manufacturers in R&D. Jane is going to talk to us about the delivery of biologicals. How do we make sure that when these, if it's a live biological, when it arrives in the field, it's thriving and able to perform in the way that we found it did in the R&D process. And then Ben's going to talk to us today about like managing the data and using the technology that we have today to help make recommendations on biological products, the same way that we've been making recommendations on chemical fertility with our chemical fertility analysis. So that's a little bit about what we're going to dive into today. And really hoping to address these pain points for ag input manufacturers. That's what we're going to be diving into. So, in this space, there's a lot of skepticism because it's something new. There's a lot of questions because biology is complex, and there's a lot of new products coming to the market. There's a lot of snake oil conversation, bugs in a jug. So how do we position ourselves as ag input manufacturers to really show and prove predictive, the effects, predictability, and how these products function in the field, so that we can differentiate our products to consumers. So those are the pain points in general that we're going to be addressing today, and that we see ag input manufacturers up against. So how do we support breaking down these barriers so that you can get your biologicals to market? So again, like we talked about, what we're going to kind of dive into today is these three different areas of bringing a biological product to market and commercialization. So of course, R&D is critical in understanding where our products work, how exactly they work, and why they increase yield or prevent pathogen pressure, so that we can then bring those to market. We're going to talk about manufacturing and distribution of the products so that they are viable when they reach the field and they do what we predict them to do and have done research to prove that they do. And then we're going to talk about the marketing and sales and positioning our products with the data that we have. So that's kind of what we're going to dive into today. And so I'll start by just talking from a biomaker's perspective on research and development. Their agriculture is, there's so many different crops out there, so many different soil types, environments, and so on. So how do we really understand how our products work so that we can position them effectively in the market? So, for those of you who are new to Biomakers, just to give you a little better understanding of what we do, we're really decoding the soil life. So we've created a standard with our BeCrop technology for understanding the functionality of soil biology. And what we're able to do with this technology is many, many things, right? But the one we're talking about today in specific is research and development and then commercialization of products. So how do we understand exactly what they're doing, where they're doing them, so that we can support the R&D process as well as the commercialization and product placement. So modeling soil functionality. This is kind of high level, the steps that we go through. So obviously we're taking soil samples, which looks very similar to a chemical fertility analysis. We're doing composite samples, and we don't need much to look at the biology. We extract the DNA, we look at the biodata, and then we use our database to generate functionality reports over time. So this is how we are gaining the insights to make informed decisions on biological products, both in the R&D process as well as commercialization. So again, we're looking at the various microbial communities, what they're doing, identify different biological pathways, identify pathogen pressure, identify stress adaptation and resilience. So the things that we're looking at when we're looking at a product and how it performs in the field are a few primary categories. We're looking at stress adaptation, we're looking at hormone production, we're looking at nutrient pathways. So how is biology moving and grooving to move nitrogen, phosphorus, potassium, a handful of micronutrients like magnesium, zinc, iron, calcium, and so on. How are those things being mobilized and changing over time when we're looking at field trials and how products are performing? And then we're also looking at pathogen risk and pressure, which is not just related to the abundance of a pathogen, but is related to many factors like the ability for the soil to cope with stress, or the beneficial microbes or bacteria that are there to balance out a fungal pathogen. So we can look at all of those things when we look at the soil in this way and understand really how products are changing and what application rates are ideal to address the issues and the intention of our products. So that kind of leads into our BeCrop trials and what that looks like. So we partner with ag input manufacturers to really help them understand exactly what their product is doing and why. So maybe you already know what your product is doing, but you want to validate that it is doing that for sure. And in the process, we often find other benefits in addition to that, which you may not have known about because you designed a product to do one thing and it has these other intentions or purposes that we can discover. So what a trial looks like is, like I mentioned, so there's these various things, disease risk evaluation and management. We can look at yield projections and improvements based on what the biology is doing. We can look at different variables that impact input performance. So like I mentioned, how your product is evolving and changing in different, or how it's working in different soil types or with different cropping systems, so that you can really understand product placement, effects across different locations and over time. So whenever we run trials, they're replicated field trials. They're with a control to compared to an applied, at least replicates of 3, so we can really create some scientific rigor. And then we look at them over time so we can understand, based on different phenological stages of the plant, when is the best time for this product to work, how is it working over time, and so on. So again, our trials are really looking at, control to a treated, and with replication over time, so we can really understand how products are performing and exactly what they're doing. So I thought maybe we're gonna hop, one more thing here before we hop over to Jane and like the delivery of products, is what shows up in the results. So when we run this replicated trial over time, we kind of highlight the notable effects or trends of the product. So maybe you've designed a product to mobilize potassium and phosphorus because that's a key concern in your area or cropping system, and you've designed a product to address just that. So hopefully that's what we find the product to do, and those will be highlighted in the beginning to say, this had a 70%, this product created a 70% increase in the mobilization of potassium compared to the control field, which is huge. And we see that happen often with biological products. So, excuse me, the evolution of, those nutrient pathways is huge. We can look at changes in hormone production. So maybe you're working with a biostimulant, and that biostimulant targets, is targeting hormone production, stress adaptation, and so on. We'll be able to also measure those things. And then looking again at disease risk, biocontrol activity, and biodiversity, soil quality overall. All of those are factors that we're looking at and identifying and measuring the change in those functionalities over time. So what this, allows you to do is, really understand in the R&D process what your products are doing and how they're doing them. And then ultimately the goal is, right, to use this data to commercialize your products. So what is our, using the trial results in our marketing material, or over time when we have enough data to really understand product placement, which Ben will talk about later, how we use this data to create recommendation engines and pull a BeCrop soil sample to understand where a grower's biology is today, and then position our products according to that to address their exact concerns. No different than when we look at a chemical fertility analysis, how we are using the chemical fertility analysis and that data point today in the fall or the spring to say, this is from a chemical and mineral standpoint what this land needs. Now we can do that from a biological standpoint and position our biological products with less questions overall, which I think this is kind of what the market's been waiting for, is how do I place these biological products and prove to a grower and to my clients that it's actually doing what I say it does, and how do I position those based on what's needed today in the biology. So that's about our trials and looking at R&D and bringing products to market and where Biome Makers fits into that puzzle. And I will pass it on here, to Jane to talk about manufacturing and distribution of these biological products. Once you know what they're doing and where they're doing it, how do you manufacture them and get them to market in a viable way? So Jane, I'll hand it over to you. Is Jane muted?
There we go. Okay. Can you hear me now?
Yep.
All right, perfect. Thanks, Meri. And if you want to go to the next slide, I'll do a quick introduction to 3 3-bar biologics. And, you know, really 3-bar, we're what's considered an early stage company startup. We're based in Columbus, Ohio, and really from the beginning, our focus was to develop new technology to improve the viability of living microbe products being used in agriculture. So when you think about, I think Meri talked about the snake oil and this perception that these products aren't living or are not high quality. You know, that's something that as an industry we've been working really hard to overcome, and that's really where our focus begins, is making sure that that product we place in the hands of the farmer does in fact include living, highly viable microbes. And so that's kind of where our story begins. Our business model is a CDMO, which is similar to what you might find in the pharmaceutical industry. We're heavy on the D, so development of new microbial products, and also we're a manufacturer. And so we work with other companies, really ranging the whole business spectrum, from far upstream companies that are doing discovery research and have new microbe technologies that they're looking to get to the market, all the way downstream to distributors who may be looking to expand their biologicals portfolio and want to include microbial technologies as part of their products. And so we bring together kind of a unique mix of expertise. We have deep expertise in microbiology, in understanding what the microbes are doing, as well as engineering to be able to develop new innovations in technology and packaging to protect the microbes and ultimately interface the packaging with application technology to better deliver the microbes. So we cover a pretty wide range. If you go to the next slide. And really for us, it starts with the importance of viability. And like I said, when you hand a product over to a farmer, you want to make sure that they trust that the product is going to work. And so what has to happen is those living microbes that are in the product have to be highly viable and at a high concentration to be able to perform and consistently perform in the field. And what I mean by, you know, being at a high viability, you know, when the microbes are delivered in the field, typically if we use row crops as an example, they're going to be delivered with the seed at planting, right? Either as a seed treatment or as a liquid in furrow. And those microbes are going to be in and around the seed, and they're going to be introduced into an environment that may be unfavorable to the microbes, depending on the temperature, the level of moisture, pH, in that soil solution around the seed. And they're going to have to sit there for a period of time. There may be other microbes there that they have to compete for resources. And until that seed actually starts to germinate, which may be a number of hours or even days, those microbes have to survive. And you want to have a high number there surviving so that by the time the seed germinates and that root system starts to emerge, then those microbes can proliferate and grow rapidly and colonize that root system. And so that's the key to these living microbe products, is their ability to really colonize the root system, because it's in that exchange with the microbes. They're basically being fed by that emerging root system. They're being fed carbon and nitrogen and other bioactive compounds that help the microbes continue to grow. But in exchange, those microbes are providing all of these wonderful bioactive compounds back to the plant. So these could be phytohormones that are helping, you know, the root system grow bigger and faster. It can be some microbes fix nitrogen or solubilize nutrients such as potassium, like we talked about earlier, and phosphorus. And so you want them to colonize that root system. And so not all microbes are created equal when it comes to colonization. Some microbes are very aggressive growers. A good example of this is Pseudomonas. You can see the picture there at the bottom where Pseudomonas can grow very rapidly and aggressively on the root, an emerging root system. The challenge with the Pseudomonas, which Pseudomonas are found, they're one of the most heavily researched bacteria, plant growth promoting bacteria and biocontrol bacteria, but they don't form a spore. And so they're really challenging to get into a stable product and commercialize compared to microbes that do form spores, such as Bacillus, which is kind of a commonly known microbe used in agriculture. And a lot of reason behind that is because they're easier in a sense to get into a stable product. And so there's trade-offs, but recognize the importance, I guess, here of viability and the ability of that microbe really to colonize that root system. And those are the two things that are really important to have consistent performance in the field. If you go to the next slide. All right, and really where our story starts at 3Bar is this idea that biology doesn't travel well, particularly for these non-spore forming bacteria like a Pseudomonas or a lot of the other microbes that are currently being discovered in research. And so once a product, you produce the microbes and you package that product and it leaves the manufacturer, one of the things to recognize is that these microbes are going to decline in viability over time. So they've got to go through a number of different steps to ultimately make it to the farm to be used in the field. And as an industry, we have some solutions that we're using right now. One is to overproduce the microbes so that you're putting basically more microbes in the package than are needed to account for that loss. Other strategies include using refrigeration to try to slow down the loss of the microbes. Obviously, a cold chain is more expensive and complicates distribution. And then finally, there's expiration dates. A lot of these products can have a short shelf life. In some cases, a matter of several months, even with refrigeration. Go to the next slide. All right, so kind of where our story begins with this is, what if we can shorten the microbe's journey? And so rather than having a very complicated production of the microbes, which currently with traditional manufacturing involves scaling up that production to very large vessels, stainless steel vessels that are known as bioreactors, what if we can simplify that production and eliminate all these other steps where microbe viability is lost? And so that's where this idea of just-in-time fermentation came from, which is the technology that we've developed at 3Bar, where we're essentially pushing the growth of the microbes as far downstream as possible. And we're doing that in our live microbe package system. So if you go to the next slide, I'll talk through real quick what the technology is. And it leverages basically single-use fermentation technology that's been around for a while in the pharmaceutical industry. It's used, has been used in that setting with mammalian cells. But we're using it and applying it to bacteria. It involves a two-chamber system where we do protect the cells in a stable dry formulation in what we call our cap. So we only need a small amount because it serves as an inoculum source and it's not the finished product. That cap is attached to a second chamber, which is a plastic film bag that includes liquid nutrients and a gas headspace. And so when we activate the product, we've made it push button simple so farmers can grow their own microbes on site. The dry formulation will combine with the liquid nutrients, and then those microbes will grow very quickly. Within 24 to 48 hours, they'll go through an exponential growth cycle and they'll be ready to use. And they'll grow to a high concentration of roughly 1E8 to 1E9 CFUs per mil, which is a lot of microbes to deliver in the field. And they're fresh. So they're fresh and highly viable and they're ready to go. All right. And so go ahead, Meri.
I was just going to frame it up. I mean, this is one of the questions that we wanted to address was, how are the microbes produced and where are they coming from?
Yeah, and so when you think about kind of the way conventional or traditional way to produce microbes, if you go to the next slide, it can be a very complicated process, but producing biologicals, you have to grow a lot of microbes. And traditionally, this has involved these large stainless steel tanks. And there's process and intent. Industrial fermentation by originally was that the microbes are going to produce something, whether that's a chemistry or a drug or an enzyme, and the microbes themselves will actually be thrown out as waste, and then the bioproduct downstream process. With living microproducts being used in agriculture, how your industrial fermentation processes undergo, there's a lot of risk for contamination during the step-up process, and capacity is an issue right now. You know, investing in these large stainless steel facilities can cost, you know, tens of millions of dollars.
So how is 3Bar's approach different to biomanufacturing in general? Because I mean, this is, when we first talked, it was something that I'd really never heard of. So it's exciting to hear that there's new technologies out there to help bring these things to market.
Yeah, so, you know, we really started by rethinking how to produce the microbes with this idea of pushing it, you know, the growth of the microbes as far downstream as possible. And that helps obviously with, you know, you're going to have a consistent, fresh, highly viable population of microbes each and every time. What it also presents is a new way to manufacture these products where, you know, we like to think about it as, you know, you get out of the tank, right? You're not tied to the logistics and the complexity of growing these microbes in large stainless steel vessels. Instead, you know, we can make the dry formulation in advance and store it under refrigerated conditions or low temperatures. But then for us, the manufacturing, rather than growing microbes in a large tank, it's instead a very modular and assembly-based manufacturing approach where we are assembling the packaging real time. So we can shorten, you know, we don't have to have long lead times or long forecasting. We can produce the microbes and the dry formulation in advance, and then it's really an on-demand manufacturing process where we can assemble the packages and then send them out the door.
So just to kind of clarify and frame up where we are so far, just to make sure I'm following, and hopefully everyone else is too. So you have this, you know, the dry formulation that's in this little cup here, and then you would sell the product with the dry formulation up top and the food for the microbes on the inside, and then the grower, or like there's instructions on the label that says to push the button 24 hours ahead of use, kind of thing. Cool. And what does this look like? Sorry,
yep, that's right.
What does that look like at scale? So we saw like the gallon jugs, but do we have this in like tote size applications?
So the great thing about using the plastic film type of technology to grow the microbes in is we can scale those bags, yes, to tote sizes. Right now, our product is based on what can physically be lifted, so up to about 20 liters, between 16 and 20 liters, which is, you know, fairly large size, but it can be larger, yes.
Awesome, thanks. So yeah, curious question, like how are these microbes living without oxygen in these bags?
Yeah, and if you go to the next slide, there is, hopefully we can get the video to work. If you touch at the bottom of the screen and press the, oh, it might, we might not get it to work real time. There should be an arrow at the bottom, maybe not. Anyways, I'll talk through it. What happens is, you know, the microbes are growing with a low level of oxygen, different than what they would in a bioreactor where you're providing a high level of oxygen. So it's not as efficient of a process as in a bioreactor, but they are still able anaerobically and through fermentation to multiply and grow. So we get to a high concentration of microbes, even though there's a low level of oxygen present.
And so are these freshly fermented microbes performing better in the field?
Yeah, so we've done some field trial work, which you can see here. I know it's a lot of numbers, but to show you, these were replicated small plots done with the university over, it was 1 year's worth of data, but over a number of different locations. And on average, we were able to show in comparing to a check plot where there was no biology applied to a commercial fermentation, where this was one that had been manufactured upstream and commercially formulated. And we took that same microbe and then we produced it using the live microbe system. and compared that to the commercial formulation. And you can see in the data, on average, we were about 3.7 bushels per acre of corn higher than the commercial formulation. So we do believe there's something to be said about having those microbes in a fresh, highly viable, freshly fermented form that can contribute to more consistent, better performance in the field.
Awesome, thanks, Jane.
Yeah.
So yeah, it's awesome to hear just more about the manufacturing process and how we can really bring high quality biologicals to the space. And so next kind of is, so we've talked about research and development, we've talked about the marketing or the distribution and manufacturing of live biologicals, which there's live biologicals, there's biostimulants, there's so many different things, but that's addressing how we bring viable live microbes to the industry is really critical and important. And we saw some really innovative stuff from Jane, and so now we're gonna pass it over to Ben to really talk about the marketing and sales. So all of these processes, the research and development, the manufacturing and distribution, and then ultimately the commercialization are all parts of this supply chain, right, to bring these products to market. So want to pass it over to Ben to talk about what FarmQA does and how we're working together to position products.
Excellent. Well, thanks, Meri. Yeah, so just to get at a high level, FarmQA, we're kind of on the data management side. And so once that product does hit the field, we're helping that agronomist, the grower, make the decision of what biological product should I choose, what should I apply, and at what rates. And we're doing that through data management. So providing and serving up data, whether that's the soil results side by side with tissue examples or tissue results with the BeCrop test data, and then putting that in a visualization that helps make a best decision. So that's kind of in a nutshell what I'll be walking through. But I do want to take a step back and just lay out the baseline functionality just to give a frame of reference for some listeners here. FarmQA, we were started in 2015, and we are a digital tool for agronomists is our tagline. And so really what that means is we're a software platform with a mobile app and a web view. And our functionality is an easy to use crop scouting application, which with treatment recommendation functionality, whether that's insecticide, herbicide, fungicide, or biological treatment recommendations. And with that, you can write those treatments in real time, or you can do it in a planning phase where you get rolled up product totals for how much you might need in a coming year. So that's where kind of the sales and marketing comes in, is looking through making purchasing decisions based on data. And with that, we roll the data up into what we call analytics, where you can see comparative charts, if you will, across whether that's your soil data, your scouting reports, maybe it's your imagery data, your yields, and comparing that side by side. And so if you go to the next slide, I'll walk through really what that looks like in a more practical example. So on the next screen, you should see our scouting application, where you've got on the left-hand side, we've got customizable forms. And so when we say scouting, this is allowing an agronomist, a grower, anybody in the field to build out what data they want to capture. And so think of this as you're painting the picture or attempting to tell a story of what's going on in the field throughout the course of a time period. And so you're tracking through pictures, through numeric assessments, what's happening. And then you can generate a report like you're seeing on the left, which can be texted out, it can be emailed, viewed electronically in our platform, and stored over time for a producer alongside a treatment recommendation. So on the next slide, getting into more of the details, you'll see we start on a kind of a map-based program where you can manage your crops, your varieties. You can see your location for where you're in the field and you're making these assessments. So in that middle screen, you can see where you can then overlay any really spatial data sets, whether that's imagery, it's yield data, it's EC maps, and that's going to help direct your efforts when you're physically in the field on our mobile application to try to identify where there's perhaps differences. Maybe a biological was applied in a strip trial. You can physically walk out and you can assess before and after or side-by-side information, which you can see on that right-hand panel where you can get into those customizable forms and start taking pictures, doing assessments, whether it's numeric, whether it's pick list, whether it's text-based. From there, if you go to the next screen, you can then immediately write a treatment recommendation from the mobile app. And so if you write a recommendation, The middle screen shows how you can then pull up a customizable database, whether that's biologicals or it's a combination of biologicals alongside commercialized products. And you can combine those into common tank mixes so that when you're thinking about writing a recommendation, you can simply nickname these mixes, search for them, find them, and then pull that in on the right screen into a kind of a finalized product recommendation where you can see rates pre-populated. They can be edited and updated along with pricing information. That agronomist or grower can view the labels or any sort of notes they've documented about efficacy of certain products. And this can all be written as part of a batch. So if you are, let's say, working with 5 or 10 cornfields that are all going to be treated the same, you could multi-select the fields, select your mix, and then choose to apply that from the mobile application. And then if you go ahead and go to the next screen, the report or the roll-up of that data for the producer is then field by field breakdowns of what should you apply at what rates, and then we're going to total that up based on your field size and the number of fields included in that treatment recommendation. And so with this is going to include biological products, like I said, alongside adjuvants, fertility products, insecticides, etc. And so it's kind of all encompassing and flexible to account for those different products. But what you're seeing in this report, like I said, so this is a batch recommendation of 2 fields. But if you actually look at the bottom in the green box, that's where I call that the shopping cart. So we're going to roll up at a report level or within one field how much product is needed, calculate it out. But it also ties into our planning functionality. So when you think about if you write futuristic recommendations, you then get a roll up across a season of how much product you either used in a season, or looking into futuristic years, knowing how much to purchase, let's say in the spring, to make sure you have the planning, the budgeting done. And so the tool then is to say, all this is being captured digitally. So you have the opportunity to look back and start to quantify what was the benefit of applying product X, Y, or Z relative to, let's say, your soil results or BeCrop data. And so this is kind of the first couple slides here, walking through how you capture data, how you can paint this story of what's going on within a field. But then if you go ahead and click on the next slide, this is where the data starts to roll up. So we have what we call analytics. It's really where you're comparing, well, it's the scouting data, maybe it's side by side with your weather information, GDUs, precipitation, or it's your uploaded BeCrop data side by side with tissue information or soil data. And within our interface, we give that agronomist or the grower the levers and the dials to turn to generate the comparative graphs that help the grower understand the story of what's going on in their fields or across a larger roll-up area or region. And so this is just one example on the chart, and we'll talk through some biological specific scenarios. But within this, you generate the comparative charts, you can push them into a dashboard, but really a large part of our value is the communication or two-way pipe between a retailer to an agronomist to a grower, and having the ability to share that data across organizational units. And so within FarmQA, you can actually white label the software, you can bring in data from any sources, but then the key thing is the growers get access to see the information just at their farm level. And so there's the data security piece around the information, but with really the crux of this is making the data more consumable, if you will, and allow you to make better decisions. So that's baseline functionality. We'll get into how we specifically work with biologicals. So if you want to go to the next slide, what I'm going to show here. Oh, go ahead.
Or I guess, well, Ben, I was just curious, maybe you're going to get to this, but how can you just lay out again how exactly FarmQA is helping with the marketing and sales process of biological products in specific?
Absolutely. Yep. And that's a perfect question. So if you go ahead and go to the next slide. So where we're helping on the sales side is more about how do you quantify the value of a product? And so I think what we provide is the ability to agnostically bring in data. So whether that's from on the left-hand side, you're seeing a lab result, a soil lab result that was perhaps collected in the fall, processed through a kind of a traditional chemistry lab, versus in the middle, you see a BeCrop test with the results that Meri was talking about, whether it's assimilation of nutrient levels. And then on the right-hand side, you see a tissue report from, let's say, Midwest Labs, where you're looking at what is actually assimilated and taken up by the crop and tested in, let's say, the tissue results. So what we're doing here is allowing you to bring that data in, visualize it, compare it, tell the story to the grower to then make a more informed and better decision as far as what product to recommend. And so if you go to the next slide, this is kind of the culmination of that data. And so what you're seeing here on the left-hand side, we're going to help you with the collection. So where you are physically in the field, you're collecting those soil samples, you're collecting tissue samples, and being consistent with where those are captured. So geolocating and returning year over year. But now once that data is captured and results are collected from the lab, we can bring those in from really any source and start to compare those. And so if you look at the dashboard that I'm displaying here, the top two graphs show calcium assimilation and iron assimilation. And really the crux of what we're talking about here is the top line, or the turquoise line on the calcium graph on the top left, is looking at the soil data. So you see the results, and this is actually over a 3-year period. So there's 3 results over 3 years, and we're looking at long-term trends of the application of a biological product. So what you see is the top line, high soil levels. In the kind of the middle blue line, that's low tissue results. And then at the very bottom, that black line is projecting on top of that the BeCrop test results over a 3-year span. And to summarize what we're looking at, the BeCrop test results are showing low calcium assimilation levels, meaning that that plant is not able to consume the calcium that's in the soil, so that we're seeing from that top line. Now, as you apply a biological product, there's a red dot at the bottom. In the actual interface, you could click on that and you could see what product was applied and when. But the general concept was a biological product was applied at that point. You then see an inversion of the data. So the BeCrop test shows higher assimilation results, meaning that that plant opened up the biological pathways for that plant to consume that soil data, or that calcium that was high in the soil. And then you see the response in the tissue data where that result goes up as well, showing that before the plant was kind of locked out from tapping into that calcium, a biological product was applied, and then you can see kind of the immediate response of that plant of being able to consume that previously kind of locked up calcium. So with that said, this is where you can see that data, you can tell that story to a grower, you can quantify the value of that application by looking at the change in assimilation rates and actually nutrient rates across different sources. So now if you go to the next screen, the kind of the culmination of what do you do with this data is you make better treatment recognizations.
Ben, can I pause and ask you one quick question here? Or
Absolutely.
Just kind of,
yep, fire away.
Because this is really, like, here, let me turn my screen on so you guys can see how excited I am. This is really, really cool. Because, Ben, what we're doing with growers all the time is sitting down with all of their reports side by side and trying to get them to see this, right?
Exactly.
That when you look at just chemical fertility, there's enough calcium here. You've got plenty of it.
Exactly.
Right? But what we don't have is the biological pathway, and that's why the tissue sample is low relative to what we have in our soil. So like seeing it all in line helps so much to visualize what is going on. And then what you're talking about is, so we introduce a biological product right after we diagnose the issue, right? So we've diagnosed the issue sometime here, and then we introduce a product, and then you see that the calcium pathway, the biological pathway, is going up. I'm just reiterating what you said, but like it's just so cool to see it on a graph. The calcium pathway biologically is going up, which is actually allowing the calcium to flow into the plant tissue and use the calcium that's in the soil.
Exactly.
So this is a biological product introduced here, not more calcium. We have enough calcium. So when we can diagnose that there is a calcium deficiency because of a nutrient pathway or lack of biological activity, then we can go in and treat in season. And even maybe we diagnose this before, this is early on planting. And, we know ahead of time that we're going to see calcium deficiencies and can introduce biological. So sorry to take that away from you, but I just wanted to reiterate what we're seeing here. it's super, super powerful, and love that you guys can show that on one graph.
Yeah.
So
Well said, and you nailed it. I mean, that's exactly what I was trying to illustrate there and the scenarios. And if you think about from a producer level, you look at 3 different reports side by side and over 3 years, now you're talking about you got 9 reports in front of you, and you're to some extent you're glazing over and you're kind of missing what's going on. And so, we talk about, maybe we're data nerds, but you start to look at this and there's value in just kind of the visualization or the ingestion and the visualization of the data in this way, to present it back, to tell a story, to then, yes, get to this slide where I'm kind of walking through the stages again on the top left, where you've got a list of all the biological products that you have at your disposal. And you start to, so this is customized to what you have access to. You can document within kind of this form of what the efficacy was over previous years, the product labels available, but then you can flow that through to kind of the bottom right, to the mobile application, to writing the recommendation, where you have an actionable report for a producer that he can send to his field hands. He can send out to a sprayer to enact this process. And so it's kind of start to finish the flow of, here's the data that helps you make the best decision with a list of the best products, which then results in a very actionable report to a producer. And the final slide I'll talk through is, again, just illustrating, then it's all about data transparency. And so on this slide, I'm showing the data is stored over time, and so it's not like you're putting it in a dusty folder in a filing cabinet and you're not going to look at it again. The data is here, so as you continue to tell this biological story, whether it's through those scouting reports and taking pictures weekly and seeing the development of the crop as you make this application, or it's your lab results going from soil to tissue to BeCrop, or even SAP test results, the data is accessible to the agronomist, but more importantly to that grower as well. And if you tap one more time, there should be a slide that shows, we also then have a journal where it's going to roll up the data on that left-hand side, where you can see every event that happened in a field, whether it's when that crop was planted, when it was scouted, a treatment recommendation, whether it's biological or as a fertility product, to a harvest date with the yield amounts. So this is a ledger, if you will, of everything that happened in that field, which you can then generate and report, let's say for maybe carbon premiums, or if you're just looking back of what did we apply in all of our fields and what was the yield differences. And so that's the power of a software tool like this, is that the data helps make those sales and the marketing decisions for a producer by visualizing the data and showing the trends over time. So that's FarmQA in a nutshell, and we'd love to chat with anybody who wants to dive in more.
Amazing, thanks, Ben. So we've got 10 minutes left and we have a handful of questions. I'm going to get through as many as I can. Maybe really quick, I'll just go back here. I'll start here maybe. So just to kind of like sum up how some of these technologies flow together, right? Like how, this list of biological products here is based on the product portfolio that you have as an agronomist. And you have probably, hopefully you've done some research with Biome Makers to understand what the products actually do and how they do them and where they do them best. And then when you have this sample and we can look at that there's a calcium deficiency in the soil based on, or there's not a calcium deficiency in the soil, there's a biological deficiency in the calcium mobilization, then we can start to put these things together. So just this illustrates really well how having the research, knowing what the products do exactly, and then when you pull the product, being able to use the right product in your portfolio to address the issues at hand. Super valuable, awesome stuff. Let me get in here to some specific questions. So we have, let's see, Andrew Ayers, is Biome Makers planning on looking at all of members of microbiome, including microalgae? So we work with a lot of microalgae clients and product manufacturers. So we work with biostimulants, we work with plant food, we work with live biologicals, anything that is stimulating or working with the soil biology, we can look at the effects. So we don't identify microalgae itself. What we measure is the how the microalgae influences the soil biology. So algae, kelp products, so on, are usually biostimulants They usually stimulate, we see hormone production by bacteria and fungi and stress adaptation. So that's where we see those products thrive most of the time. And we can measure the changes in the biology and the bacteria and fungi that the algae attracts. I don't know how that part works, like why that happens, but that's what we're measuring on a biostimulant side of things, is exactly, you know, those products are coming in and how they're influencing soil biology. So hopefully that answers your question. Feel free to follow up and reach out to me directly if you want to dive into that more. This question is for Jane, wondering how the farmer confirms that just-in-time microbes have hit the desired concentration before they're applied.
Yeah, no, that's a great question. Well, we like to have the physical indication, right? The visual indication. So farmers can see that they've grown their own microbes because it's changed to a very turbid, cloudy visual. So they know that they're fresh because they saw that they grew. We give a timeline based on our testing, and usually we recommend that it's at least 48 hours. Depending on the microbe and the conditions, temperature conditions. So we let the farmers know to wait 48 hours, but they should visually see that the microbes have grown because it's now cloudy and turbid instead of being clear.
Okay, awesome. So there's a visual idea. And I mean, once those biologicals are delivered to the field, as long as there's something growing in the field and something for those microbes to eat, they'll continue to grow once they hit the field also.
Right, and that's key. That's key.
Totally. Awesome. Thank you. This is a question for Ben. How do you determine the recommendation product or brand? Is there a database that you will select product candidates? Maybe, so it sounds, so what I'm hearing is like for, you know, in the database that you had listed the selection of products, it sounds like you select what products you want to be in your portfolio to pick from to make a recommendation.
Yeah, that's right. I guess maybe I'll answer this in two ways. You're right, Murray, in the sense that a customer that uses FarmQA can define, here's the products that are in my database. We also do have like a CSV import to say, I just want all the biological products that are known to the industry. If you'd prefer that method, we could bulk upload, and then you've just got this exhaustive list that you could search. So that is an option. If the question was going more towards, does FarmQA kind of automatically say, based on these results, you should apply X? You know, we don't have that algorithm in the system, so it's more about looking at the data and that agronomist understanding, okay, based on this data, maybe these are some good options.
Yeah, and just a a point there too. You mentioned you don't have the algorithms yet to like actually generate a product recommendation with like computer learning or artificial intelligence. And on the Biomaker side, that's something we're working on. So as we collect more data from product manufacturers and we partner with ag input manufacturers on the research and development of their products and commercialization, once we have enough data from, with a certain product, we can use artificial intelligence to position a product based on the polling of a BeCrop sample. So that's where, the industry is headed. If you're curious to learn more about that, definitely reach out. But I'll just say that that's what's coming. And I think that, I guess that's what's coming in the industry, is to have enough data on biological products to be able to make those recommendations automatically. It's what everyone's asking for. So it's just who's getting to the punch first to have their products well researched enough to really use that type of technology to position products.
And I'd quickly just throw out too, we're positioned where we could bake in an algorithm like that through a partnership, where we could have that built into our engine based on the algorithm that has been developed. Okay,
awesome. Maybe Ben, this is kind of, well, let's see. There's a lot of questions here and we only have 5 minutes left. 3 bar. Have you thought of implementing personalized medicine to the field, possibly using a soil sample from a specific field and creating a personalized prebiotic solution?
Yes, that's a great lead-in question. And I think you guys just kind of set up this idea of being able to diagnose and prescribe. And for the input manufacturers, our ability to be flexible to create products that are maybe stacked, or the flexibility to use specific products that are kind of that medicine, personal medicine type of approach. Yes, that's where I believe with precision application and being able to package things in a way that it uniquely can interface with those precision application tools is the wave of the future. And we've seen some of that start with fertility, right? With variable rate application and prescriptions, and biologicals is going to eventually be there too. It's going to take time. But yes, I think there's a lot of entities out there that are working towards that goal.
Yeah, I see so much potential there. You know, we get that question a lot too, is kind of like, you know, we're the diagnostic tool, right? We're the diagnostic tool that says what you're missing or what needs to evolve or change to open up your calcium pathway. And so being able to have a company like 3Bar that, you know, you have all these little cups on the shelf, and one is for calcium mobilization, you know, has bacteria to help mobilize calcium, and maybe there's a, you know, algae. I don't know if that can grow in there too. Maybe they grow in tanks also. Go with me here. But like so you need some biostimulation to like help move and increase stress adaptation. You also need calcium mobilization. Well, you got your BeCrop test result, and then you can go and like custom select, I need these 2 little cups, and then I'm gonna grow them and apply it to my field. Like, it's such a cool technology to actually make that viable. Really, really neat stuff. And I can imagine that like plugging in then to FarmQA, where you have it all in front of you, and you know, you can go to 3Bar and select exactly the custom layout that you need. So really,
right.
really.
I was going to mention too is that with that, if you collect samples of multiple spots, we can generate zones and do variable rate application maps. And so with that, if you got to the point of doing multiple BeCrop tests, we could then handle on the other side of attempting variable rate on biologicals.
That's so cool. Let's see, there was one more question which I think would be great to address in these last couple minutes. I'm not sure who it came in from, but the question was, where'd it go? How might a grower think about ROI in the calcium example? Maybe we can go back to that, and then we could kind of tackle this one together. But I'll give it a start if that's okay.
Yes, please do.
Yeah, so I mean, look here. To look at this again, there's various you know, metrics here on the y-axis, and so it depends on what test you're looking at, I assume. Yeah, right. And these are all relative indices, right? So in this example, we have identified using a chemical fertility analysis or total digestion. I'm not sure which is in this particular one, but we know that we have a lot of calcium in the soil. We have enough calcium in the soil. When we looked at our tissue analysis, the plant is not getting the calcium. Why isn't the plant getting the calcium? Because the biology in the soil is not active in that pathway to actually mobilize the calcium and get it into the tissues. So that's our issue. So you could go in, and maybe if you just took this tissue sample, then you're gonna, you know, pay for a foliar spray to bump up that calcium from the top, which is fine. That's one way to get calcium into the plant when you need it. But if we address the root cause by introducing a biological that helps open up this pathway and move the calcium in the soil, we're not paying to add more calcium when we already have enough calcium. What we're doing is looking to nature and identifying what needs to happen in the soil biology, because as long as we keep nurturing that biology, it's going to be there and it's going to help us continue to leverage the calcium that we already have. So that's where the ROI is, you know, is that we can learn and understand why we're not being able to use what we already have and address the root cause. And, you know, by using root cause analysis and addressing a root cause, that's going to have longer term impacts for us in the future, rather than just treating the symptoms. Now, we need to treat symptoms sometimes, that's what we need to do, just like in medicine. Like, sometimes you need to address a symptom immediately and then work to address the root cause. But that's where I see the ROI coming in. I thought that was a great one to end on. And please reach out if you have more questions to understand that. All of these technologies are really powerful in just helping us understand soil and soil ecology and soil health better, so that we can produce ROIs by looking at the soil biology to help us leverage what's already here and really understanding what's going on. So I'll end there. I'm sorry we didn't have time for all the questions. There's a lot more. We will reach out to you directly to answer unanswered questions that we have this week. And please, again, reach out. All of our contact info should be in the follow-up email. You'll be able to find the recording of this webinar to look back at things if needed. And we really appreciate all of you being here to chat with us about bringing new and innovative biological products to the market. Thanks.
Thanks, everybody.
Thank you.